Talk is essentially, a story of collaborations between multiple groups, and I'll mention each group as it becomes relevant. But essentially, we are talking about this because Avinash and Alka, who are the graduate students who are now graduated, were roommates at some point, and this is study basically can be linked back to them being roommates and discussing how their different research interests intersected in this, resulting in this collaboration. Right. So why you want to understand or study this is because cell migration is important for various physiological and uh, pathological conditions. And what is interesting is that uh, the cell migration is it can be tuned, the mode of cell migration, depending on several factors, but more importantly and relevant to this talk is the ECM architecture, right? And in typically, it is quite difficult to control ECM architecture in a reproducible way, and also do the imaging of the underlying ECM uh, matrix and to learn about the cell migration and what forces they are exerting on their environment to achieve this kind of migration. So to partly address these limitations, one approach which is the basis for this study is developed by Dr. Amrindan Nain at Virginia Tech through his StepLag. They have a technique where they can generate these nanofiber scaffolds of nanofibers of hundreds of nanometers in diameter, and they can precisely control what is the spacing in between these fibers, right? So essentially, you can think of these as nets formed, for instance, by spiders to have a visual image in your mind. So these are 2D networks, and you can coat them with fibronectin, and you can culture cells on top of this. So what this allows us to do is to understand cell fiber interactions in a very controlled way, where we can reproduce the underlying matrix, and it's a structured matrix, so that removes the some of the variables that we cannot control in other cases. So using this platform, they have already shown several interesting ideas and observations, but what I want to highlight here is that changing the spacing of fibers can capture how cells behave on or respond to different fiber architectures. And what is basically seen is that as the fibers get dense and dense, the cell behavior is closer to what we'll observe on 2D flat surfaces. But as the fiber spacing increases, then the cells behave, of course, differently. And in cases, it is it reminiscent of how cells interact in a 3D fibrous environment. So the question that we are trying to answer in this study is based on, this is what a typical experiment which were performed in this lab would look like, right? So we have the underlying fiber matrix. These are polymer fibers. And as the cells migrate on top of these fibers, of course, the cell-exerted forces will deform the underlying fiber. And the question that we are trying to answer is, how can we infer the forces that the cells are exerting in order to create these deformations? So the problem seems, it's not a new problem in the field of biophysics. People have done traction force microscopy on soft substrates, where you'll have to embed in the soft substrate some fluorescent microbeads, and tracking these microbeads will allow you to track the deformation of the underlying substrate. And from the deformation and knowing the properties of the gel, you can extract the forces that must have caused that deformation. So the idea is similar, but there are some inherent challenges which I'll explain that come about since we are talking about this fiber grid. First things first, we need to extract the deformation, and having a structured grid really helps to extract the deformation directly from these kind of images. But to get the fiber images, we took advantage of machine learning. So, I mean, the obvious way to go is to label the fibers and image them. But to circumvent this step, what we did was we trained a neural generative adversarial network in collaboration with Dr. Karpatny at Virginia Tech. And what they did essentially is took the phase input image, took the ground truth, which told us about the location of the fibers from the fluorescent images. And with enough examples, we can get the images we can extract the fibers directly from the phase input images without having to go through a fluorescent labeling of the fibers. Once we have these images of deformed fibers, we can extract the deformation by tracking where the intersections of the fibers are going. So you can either do this by hiring an army of undergrads and asking them to label everything, but I think a better approach would be to again use that training data and build a network that can do it for you. On top of that, you can make sure there is a consistency maintained between tracking of the nodes and all of that. And you can also enhance it a bit by using some sort of template matching, since the fibers form like a plus sign at every intersection. So we can get all of that. And what we have so far is we start with this phase image from which we can extract the fiber shapes using this deep learning approach, and we can track the intersections of the fibers. And since we know that the fibers before the cell was put on them form this closely or nearly orthogonal grid, we know the reference shape, so we don't have to do anything to to remove the cell from the substrate to get the reference fiber locations. We can get them by using the fact that the fiber grid was orthogonal. So using that, we get the deformation field. And the question I want to focus on now is how do you go from this deformation field to the forces? The problem is this going from deformations to tractions or forces exerted by cells is ill-posed because of the essentially Saint-Venant's principle, which tells us that statically equivalent forces can produce the same deformation far away from the point of application of the forces. So that's one issue. The second issue we are facing is that we don't have the measurement data at everywhere on our grid. So we know that the fiber intersections, how they are moving, but we can't really track individual material points on the fiber itself because we aren't, tagging. We aren't using any specific markers for a material point on the fiber. So all we have is the overall deformed shape of the fiber, not where each material point is moving. And on top of that, the mechanics of the fiber network is highly non-linear because the amount of forces that the cells are exerting causes the fibers to go beyond their linear Euler-Bernoulli kind of response. So we need to account for the nonlinear mechanics of this network. So how do we solve this problem? We pose this as an inverse problem. So the knowns in this problem are the displacements at the fiber intersections, and unknowns are what are the forces or tractions T exerted by the cell. So we pose this as I'll explain each term, but we pose this as a constraint minimization problem. Okay, and what are we minimizing is that we are trying to match the displacements at the fiber intersections, this yellow arrows, with the displacements u that is created by the unknown forces T, which will apply in our numerical model. So that's what we are trying to match or minimize. But because the problem is ill-posed, we cannot do this. If we do this, just this, then the noise in our measurements will affect the prediction of tractions very severely. So we need to constrain or regularize the problem by saying that, okay, uh, uh, don't penalize if you are trying to if you get very high forces. So we add regularization through this regularization parameter beta. We impose that the deformations created are not any deformations, but they should come from the mechanics of the underlying mechanics of the nanofibers, which we model as beams, co-rotational beams that can undergo large displacements but small strains. And lastly, since we have the shapes of the fibers from the images, right? What we can do is ask the solver to also try to match these deformed shapes to make use of all the information that we can get. So although we don't know exactly where the material points on fibers are going, we can ask the inverse solver to just do your best in trying to match these shapes as best as you can. And this is a technique for segmenting used typically as active contour, where you can using a contour and asking it to go to the borders of a shape or to minimas of the image landscape, it can segment things out. So we do the same thing. We get the fibers to be the low energy region and ask our solver to go there since we know the overall shapes. All right, so we implement this, and before I show you any experimental data, I would like to verify that things are done correctly. So I'll first show you some verification cases, and then we can talk about some experimental force recovery. So in a few slides that follow, on the top left, we'll see what is the force that we are simulating. So this is to generate some displacement pattern. And we'll use this displacement deformation pattern to infer the forces, which we call as recovered forces in green. And we'll plot the recovery error in the forces. And this is plotted as a function of this regularization parameter beta. The idea is that if beta is too high, then we are penalizing the magnitude of forces too much. So the forces will end up being close to zero and the errors being high. But as we decrease this regularization, as we release the constraint on the value of forces, we see that uh, uh, we are able to match the actual force pattern that was simulated. So in this simple case of a force dipole, what we see that using either the entire information about the displacement or deformation field, or using partial information only at the intersection nodes, we are able to recover the forces fully. This is not the case when the patterns get complicated. So if you simulate slightly more complex of contractile force pattern like this, which creates this deformations, then okay, so visually we see that we are able to recover the green arrows, which are very close to the red arrows, which is our target. But when we calculate the pointwise error, this is the blue curve, we see that with decreasing regularization, we are able to capture the forces, but not exactly. And this is linked basically to the Saint-Venant's principle, right? Because a statically equivalent force can generate the same deformation far away from the force application. But when we average the forces and compare them in a small radius of 20 microns, we see that the errors are less meaning that on average the forces are captured correctly, or we can also calculate the overall contractility and compare that, and that is captured very well. Then we can ask, okay, so what if we now focus only on the data at the intersections, right? Similar to experiments. And in experiments, we also expect to have noise in the measurements or the data. So we need to account for that. So what we did essentially is in the simulated displacement fields, we added some errors. 0 is no error, 0.2, and then 0.5 represents the what is the error in microns in measurements. If there are no errors, what I'm showing on top left is L curve, which is typically used to solve or choose the regularization parameter. So on the y-axis here is the magnitude of forces. On x-axis is how well we are capturing the measurements. We start at the right bottom when the regularization is very high. So if beta is very high, which is this point, then forces recovered are close to 0, and we have very high errors. As we decrease beta, we follow this curve. So what we are doing is we are trying to match the data, measurements better and better. And if there are no errors, then we see that we can match them pretty well while not increasing the forces too much. But that's not the case if there are errors in the data, like in the blue curve. After some point, the forces that are inferred will try to match the noise in the data, and that's what leads to this sudden jump in the forces measured. So the idea is that we want to be close to this L corner of this L shape, which allows us to match the measurements as best as we can while keeping the forces to reasonable magnitudes, right? And this corner in this L shape also matches the point at which the errors are the lowest, right? But what we see is that we are able to match the measurements at the intersection of fibers, but the fiber shapes themselves are not correctly matched because we haven't used the information about the shapes of the fibers using that active control energy term. But once we add that term, what we see first is that we are able to snap the fibers to the correct deformed shape. So we're using all the possible information. And interestingly, the deformed shape of the fibers introduces or constrains the system enough that the solver doesn't really like to increase the tractions beyond certain points. So we are naturally regularize the problem inherently through this constraint on the shapes somehow. So with that, we verify the approach, and here are some other examples where we can we show that we're able to recover the patterns, the force patterns quite well. And to summarize that part, so we have essentially a technique, we call it DLFM, to go from the phase image directly to the deformed fiber shapes through deep learning. Because the fiber grid is orthogonal, we can extract the deformations at the fiber intersections. And using the inverse approach, we can reconstruct the forces and measure things like the overall polarity of this force pattern and such. We have used this technique to do basic verifications like drop-in force magnitude when treated with different drugs that affect contractility. We have also tried understanding forces during cell migration on grids that are not square but rectangular, and naturally because cells have the stiffness between two directions is different, cells tend to be elongated along the direction in which the fibers are denser, and the force polarity patterns also capture this polarity of cell over the migration cycle. We can also apply this study to cell migration in other complex environments, like when cell is migrating from a square to rectangular pattern, and we show that the technique can also work well in such cases. Or when we have multiple cells interacting, we can also resolve the forces in such cases and also understand what forces are, how cells are interacting or they're dividing as shown in this example, and map the forces there as well. What is interesting about this forces, if you notice, is that if this was a cell on a flat 2D substrate, we expect that the tractions are mostly centered at the periphery of the cell. But in our all of our observations, we see that the forces are also inside the cell, which point to the fact that the cell is able to adhere through focal adhesions and apply forces not just at the periphery but also within the cell region. So with the help of a chemomechanical cell model developed by Farid Ali Safi at NJIT and Guy Jenin, we are able to show that, okay, in experiments, we see that on fibers, the focal adhesions are not are, are appearing throughout the cell region. And the chemomechanical cell model, which essentially models the contractile cytoskeleton of the cell, and focal adhesions formed, or those focal adhesions that mature as a part of the model. They show that indeed on fiber networks, the focal adhesions appear throughout the cell, unlike on a 2D substrate. So with that was one of the biophysical observations that was triggered by this initial study and the method. But I would conclude by saying that we have this deep learning enabled method that allows us to go directly from the phase contrast microscopy to the forces, eliminating the fluorescence imaging step. We included not only the matching of deflections at the intersection, but we in the model inverse formulations, we also have a way to match the shapes of the deformed fibers, which for me was a very interesting addition and feature of this model. And we are currently working on applying this technique to understanding other cell migration related phenomena that are specific to these fiber networks. So with that, I'll thank you for your attention and take any questions if you have.
All right, let's thank Sohan for the wonderful talk. Do we have any questions from the audience? You can either paste your questions in the chat or just directly unmute yourself. So maybe I can start with a couple of questions that I have. So I think, Sohan, you mentioned that the mechanical properties of these nanofibers were modeled as nonlinear. So could you maybe tell us a little bit more detail, like what are the sort of underlying sort of elastic models for these nanofibers?
So the nonlinearity I mentioned is just a geometrical nonlinearity coming from the fact that the fibers can deform a lot, but the forces exerted are not large enough to cause any really large strains, because the fibers are really stiff. These are polystyrene fibers. So we assume that the displacements and rotations can be large, but the strains in the fibers are still small. So the fiber itself still follows linear elastic response.
I see, I see. So that being said, I'm presuming also like sort of plasticity is not considered. So you don't consider the fibers deform, beyond the limit of like actually getting another sort of undeformed shape.
Well, if the forces are very large, if you put a very contractile cell, we do have instances where the fibers can, in fact, deform, in an inelastic way. But I don't fancy solving that inverse problem because that makes that adds another unknown to the mix with where we don't know how much, what is the elastoplastic model for the material. So that just complicates the story.
Absolutely, absolutely.
Sohan, can you comment on how computationally intensive is,
Oh.
trying to extract this inverse problem?
Yeah.
Is that something that you can do, like once you train the network, can you do this like in real time? Or like still you have to do like, like processing takes like a lot longer than the actual movie?
So the deep learning is only to get the fiber shapes.
Yeah.
That is very fast. I mean, once you train the model, that is in a matter of seconds to convert a stack of phase images to the fiber images. But the inverse solver is quite intensive, as you would imagine, because the underlying problem itself is nonlinear. So if the problem was linear, we can do it, in one go. But for nonlinear, we are essentially putting a nonlinear constraint on the minimization, which makes it quite intensive to solve. I think it takes a couple of hours to go through the frames of a typical movie that I've shown.
All right, thank you.
Yeah.
So I also have another quite meticulous technical question. So I think this synthesized nanofiber network, it is probably not like perfectly 90 degrees orthogonal, right? So there could be like slight imperfection in the geometry, sort of. So like in the actual pipeline procedure, do you need to like first take a picture of the nanofibers before cells are put on there, and then you put cells into the medium, and then, like, do you need to take a picture of the initial condition?
Not really. I think what works here is that if we go far enough far away from the cell, we can infer the reference position of the fibers by just connecting a straight line through the points at that are farther away. We did try using initial reference image, but I think there were some technical challenges because the substrate can ever slightly move or rotate, right? And that may cause some issues in doing in a way what you said. What we found, what worked best was to use the fact that the fibers were straight before and kind of estimate the shape of estimate the the direction and shape of the fibers based on the actual deformed image that we have.
So did you have some tests, for example, like quantifying how sensitive the result is to maybe slight deformation of that initial configuration?
I mean, yes, I mean, we have done studies on the errors in the measurements in general, and the what we see is that what we see is that for the errors that are of the magnitude of human error in assessing the locations of these fiber nodes and displacements, about 0.2 to 0.3 microns, I think the approach works very well in that for that magnitude of errors.
I see. Well, thank you.